Good afternoon. It's my pleasure today to introduce Professor Yolanda Gil from Information Sciences Institute, University of South California. Professor Gil has a Bachelor of Science in Computer Sciences from the Polytechnical Institute of Madrid and PhD in Computer Science from Carnegie Mellon University. Currently, Professor Gil is Associate Division Director of Research in Intelligence Systems Divisions at the Information Sciences Institute. She is also director of the Knowledge Technologies project, and she holds a research professor appointment in the Department of Computer Science. Professor Gil, she is primary investigator in more than 30 grants. She is an author of 9 edited volumes and an author of more than 120, as I counted, peer review articles in journals and conferences. And in addition to this, she has many leadership positions in various national and international organizations in artificial intelligence and computer science. So let me introduce Professor Gil and welcome her to the podium. And her talk today is knowledge-driven science infrastructure. Thank you.
I know you do work in bibliometrics in the center, so I guess you did some homework on that. You know more than I do about myself now. Okay, so there's a lot of interest in data. You don't have to know a lot to know that. And I think I'm here to convince you that knowledge is very important and we need to be able to integrate knowledge and reasoning a lot more in the way that we deal with data and science infrastructure. So the big message of the talk is that data-intensive computer is very important. We have a lot of data available, but there's still a lot of reasoning that you need to apply in many aspects of science. And this is becoming increasingly unmanageable, and I'll show you some examples. So this creates a human bottleneck, and my goal is to really try to focus the human attention on problems that deserve their expertise and remove more routine steps out of their concern. I think that this entire research agenda creates a lot of opportunities for people that do research in artificial intelligence, such as myself. So with this, I'll tell you that I don't have a background in biomedical informatics of any kind, but really my interest is in intelligent systems that can support scientists. I'm getting an echo here. Can people hear me okay? Yes? Okay. Here? 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 Okay. Sorry? Okay. So I'm going to talk a bit about data-intensive computing and discuss a little bit the limits that we're reaching there. That will help me motivate the need to have workflows and workflow systems. That's really my particular research area. And from there, I'll go into reasoning about workflows of data analytics and why we need semantics and reasoning. I'll show you some examples of where we've applied semantic workflows and gotten really good results. And then I'll step back and go back to what intelligent systems can do for science and talk about a new emerging area of discovery informatics. So let's start with data-intensive computing. So this is something that you all have seen. So we are in a place where you are all at the forefront of this. You know that there's more data than we have the ability to handle. You know that data is becoming so important that there's entire conferences on data citation. There's a lot of concern in the government with data management, data curation, data stewardship. So I think that the importance of data the emphasis of data in current and recent science has been very strong. Typically, when you hear about data-intensive science, you hear about a number of issues that are very important. So, but they all focus on data and computation, not so much on knowledge and on the human side of science. So you hear about data management, you hear about data movement, scaling up, data access. So all of these are important aspects, but again, they're not really the topics that a scientist would want to dwell on. You also hear about computation management, so things like reserving resources to execute your computations. You hear about collection of data, so better and better machines, or maybe sensor networks, or streaming data. You hear about resources, so you hear about cloud computing, commodity computing. You hear about virtual machines, virtualization. So all of these are very important topics, but I'll tell you that none of them have to do with what a normal paper in science deals with. These are basically means to an end. So what's really curious to me is that the bottleneck seems to be not so much the data movement, the data access, the data collection, but the bottleneck to progress in science seems to be more on the human side of science. So here's a snippet from a recent editorial in Science last year where they actually asked people on their reviewer body to respond to a questionnaire, and they asked them, do you have the necessary expertise in your lab or group to analyze the data in the way you want? And I was really shocked by these responses. I don't know if this surprises you, but really the overwhelming majority of scientists don't have the access to the expertise to analyze the data that they're able to collect. And to me, this is really peculiar, and I think that it creates a situation where there's the scientists that need to understand the data and the data analytics happening in different places. And that's kind of a curious way in which we're doing science today. I think it also says that this questionnaire went to reviewers of science. So I think that this situation might be much more exacerbated with let's say, less privileged individuals. So I think that there's a lot of aspects of science that really require a lot of reasoning and where really scientists should be focusing. So I think there's a big human bottleneck in a lot of the tasks throughout the scientific processes. So for example, a scientist may spend time figuring out what is the state of the art, reading a lot of papers, chasing a lot of pointers, talking to people to try to figure that out. What's a good problem to work on? What's a good experiment to design? What data should be collected? What's the best way to analyze that data? What are the implications of these analysis and these results? And what do I need to do to adjust my model, or what's my next hypothesis to go through? So there's actually a lot of interest in the artificial intelligence community to help with a lot of these processes. So you can see that these are really science processes, and none of them talks about data movement or reserving resources or computation. They talk about really the knowledge that is involved in the business of science. And I think that this is very exciting for us. There's, for example, systems. Larry Hunter has a system called the Analyzer that actually reads through the literature and extracts relationships between proteins, genes, different biological entities, and creates networks where he can point out possible new relationships between those entities that can be suggested as future investigations. Hod Lipson has a system called Eureka that actually is part of what he calls a robot scientist that actually runs experiments in the physical world. Sometimes they're physical devices when he's trying to learn physics models, but he's also working on biomedical domains where the system actually is collecting data, developing models, revising the models depending on what the data says, and designing the next experiment. How do I want to set up this apparatus to collect the next round of data and refine my models well? So there's a lot of aspects of science that could really benefit from a better look at not just how we handle data in science, but how we handle knowledge, models, and data analysis in science. So I talk about this because, when you talk about the knowledge to do data analytics being spread out, I think that knowledge really has to do with a lot of the pieces of what's a good problem, what data should I collect, how should I analyze the data, and what's the consequence of that. So changing the focus from data to knowledge is important. Another reason is reproducibility in science. So a lot of the data-intensive computing tools really have very little support to make the experiments and the analysis truly reproducible. So science needs to be reproducible. So I have here a clip from an article in Nature some maybe 3 or 4 years ago where the editors were saying, if you cannot describe exactly how you carried out your method, we don't even want to see your paper. Basically, our reviewers won't be able to review it properly. There's a lot of concern with retractions of papers. Last year there was a Nobel laureate that retracted papers, and not for the first time from their lab. So there's a lot of reasons to help reviewers make publications in science a lot more inspectable. So data-intensive infrastructure doesn't really help us with this. And I think that what is needed is a better way to link the data analysis that we're doing to the models that a paper is describing. I also have a clipping from the Wall Street Journal showing that pharmaceutical companies, this is an article from December last year, pharmaceutical companies taking articles from the literature on promising results that could lead them to produce new drugs, and really not being able to even replicate the results. So they're investing millions of dollars on possible future drugs that they can't even replicate the original experiment, much less develop the drug to market. So I think this is really a concern for many, and I'm sure that you all know about the Duke clinical studies where a paper was published. This was on personalized cancer treatment, and a paper was published, clinical trials got started, and they had to be canceled because the results were not as expected, and the original study turned out to have major flaws. This is work from Bayrakil and Coombs. There's, for example, sites in China and also in Romania that actually expose flaws in published results. So I think that this is a major issue. There was an issue in Science on reproducibility recently that I'm showing down there. So this is really a matter of saving lives, saving a lot of money, but also making science more serious and rigorous. So why is reproducibility so hard? If you think about it, a lot of data analysis, they're really very complex processes. They involve a lot of skills, but also they involve, and I show examples here from genomics, they involve you looking for the latest and greatest algorithms, software tools that are out there. So all of the ones listed here are openly available tools that one can use in an analysis. And so for each of them, you have to really understand how to set it up, all the parameters that they have, how do those parameters relate to your data. There's a lot of knowledge involved in setting up the data analysis correctly. And there's typically many steps and many different tools involved. So that makes the reproducibility very hard if you have a cycle of trying things out until you get the results that you're looking for. So I hope I gave you a good idea of the need to have better access to expertise in data analytics for all scientists, but also the need for better tools to keep track of data analysis and bringing more knowledge into the mix. I have a question already. Sure.
Can you distinguish between analysis done during the discovery stage and analysis done as part of the final paper? Because it seems like many of the tools that you're putting to the previous slide are ones that people use while trying to, just searching blindly in some sense, and being a bit random might be useful for finding something useful in that case. But once the result is found, the analysis that's done on that may not use these tools at all.
No, actually, so the way it might work, I mean, let me rephrase your question a little bit. The way it might work is that people have a certain type of data or a certain type of goal. They may explore different sets of tools. They may hear about new tools that have become available that might give them better results. And then once you've explored that and you have a method constructed, you publish that, and then that, that might be the one you use the next time around, right? So yes, there's different stages to research. So you might do more exploratory research when you're trying to devise a method, and other times you want it to be more operational so you can reapply it over and over. But I think also a situation that you find is that someone publishes a method and you see that they use Plink, and so you go off and you try to find that system and see how you could incorporate it into your research as well. So you have to go out and fish and learn about new methods all the time. Okay, so as I said, bringing more knowledge to capture data analytics expertise, to have a better way to capture the methods that we're using in analyzing data brings me to introduce you to the idea of workflows. And the title here says workflows are a first-class citizen in science, because I think that we recognize that data is very important in science. We recognize that algorithms and software packages are very important to science. But I think that these workflows, these overall end-to-end methods, are a first-class contribution, an object of exchange and discourse in science. So let me tell you a bit more about workflows. Workflows really represent processes. So you might have heard of workflows in the context of human activities. So for example, a patient checks into the hospital and the workflow might go something like this. They first check into with their receptionist. That produces a piece of paper. They get sent to the triage nurse. Then the nurse produces another piece of paper. You go back to the reception. They ask you for your insurance. There's a number of steps that get gone through. And often they involve people intervening in different steps of that process. There's analogous processes in science. So for example, the process by which many people go off and search for related research for a particular topic or any recent findings about this gene and so on and so forth, that involves researching the literature, and there's probably a lot of steps in those processes that are common. So these are workflows that involve human activities. There's other workflows that are more the focus of my interest, which are workflows of computing steps. So some of those steps might be services. So for example, you hear about this in the business world, so entire services of composing services from various vendors to buy a certain widget. Or also in biology, you might access a BLAST service and then go to some other service with the results and so on and so forth. My work is on computational workflows. We don't really rely on third-party services, but we really compose workflows out of computational steps that are pieces of code that we control. We have a whole library of those. They actually invoke third-party software tools, but we control where those are executed. When you invoke a third-party service, you're really at the mercy of what they deliver to you. So here's an example on the right side of a little sketch of what a workflow might look like, and it's for classifying documents. So you might take a set of documents, run some software that does a correlation scoring with all the words in the documents. You may take only the words that go over a certain threshold, so that's how you select the features, which are basically words in the documents, and then you might run that through a modeler that then helps you classify the test data. So that's, at a very high level, major steps in a workflow. Those major steps then get implemented into an executable workflow. So that's shown in the right-hand side. You can see that it has a lot of more detailed steps because, for example, you might remove from the documents common words, for example, the, of, a. And so you start to add steps that really help you implement the details. But conceptually, the left side is the overall method or protocol that you're following to do the data analysis. So I'm interested in these workflows that capture all these computations in a way that helps you see the data flow. And if you look at it, it doesn't focus on one particular software method or algorithm or step. It's really an end-to-end composition of different software tools that have different functions. And it has, it's a method for text classification. They may be alternative methods to do this, but in itself, you can run it on a set of documents and it will give you a result. And it has parameters that you can manipulate. So to describe workflows a little bit, I'll refer to a workshop that I ran at NSF some years ago where we had computer scientists doing workflow research, but also a lot of domain scientists doing research in astronomy, geosciences, biomedical research, and so on. And one of the things that I learned there is that they had this sense that everything in computing is growing exponentially, but they're really getting exponentially more overwhelmed personally. So the literature, computing power, memory, everything. So they're feeling this human bottleneck of how they have to cope with the increasingly more powerful infrastructure that they have for science. So in their mind, there's a lot of processes and steps that they follow that really could be captured and shared better. So for example, you read a paper, you try to reproduce what it did, and you really need to do some detective work and do some forensic research, which is called that sometimes forensic research, trying to figure out what was exactly done there, and that could take a lot of effort. So can we capture and share those processes? And so there was a lot of interest in really capturing in workflows what are the data analytic processes that are normally published in science, and how to make those be just as important as data or software tools are. And if you capture these methods as workflows, then you have captured a lot of knowledge that goes into developing these end-to-end compositions, these end-to-end protocols or methods, but also you facilitate reproducibility, because now the workflow system can track through these steps, can publish how the new data was created, and can have a better record of exactly how you run the experiment and how the parameters were set. And this one here is a very small workflow, but they get complicated real fast. So here's an example of how they get complicated real fast. So this is a workflow to do earthquake simulations. So the input is an earthquake forecast model, how many faults are going to rupture in the next 100 years. There's many. So if you're simulating what might happen to a particular site when you want to build a bridge, you want to see what might be the magnitude of earthquakes there, depending on how the faults break. So you start simulating different fault ruptures, how that affects other faults. There's all kinds of ways in which waves propagate through the terrain. So there's a high-level sketch of a workflow on the top of the figure. But then the workflow system is actually able to expand throughout all the fault ruptures and create more or less the workflow that you see in the middle that actually has a lot of parallel jobs, independent wave propagation executions for each of the fault ruptures, and then it's able to combine information at certain points and finally tell you what would happen on this site. So this particular workflow execution consisted of many thousands of jobs, and the execution lasted 1.9 CPU years, but the workflow system really helped create the individual jobs that needed to execute and oversee their execution. If any job failed for lack of memory, anything, it would resubmit the job somewhere else, and was able to execute the workflow end to end. So you can see that the workflow system is actually helping you manage a lot of the details that have to do with the data-intensive computing, right? The data movements, the workflow system was moving the data, dealing with failures, doing a lot of things. So there's a lot of workflow systems out there, and if you're interested, there's a very nice book, Workflows for eScience, that really gives you a range of workflow systems and their applications out there. There's, for example, a system called Galaxy that you might know, or Gene Pattern. These are for microarray and genomic analysis. There's a system called Taberna for composing biomedical workflows, biomedical services. Loni Pipeline is used for neuroscience image analysis. There's UIMA, which is a system created by IBM to do text analytics. Sorasks helps with social network analysis, algorithms and workflows. So there's a lot of workflow systems out there, and they have different capabilities depending on their needs and their resources, but they do a lot of things like, it's not just composing those nice data flow diagrams that I've been showing you, but they really manage the execution. So if you're, running a lot of analytic steps and you use a scripting language, when one job in the middle fails, you have to find some way to recover and figure out what is left to execute. When you have to move data, you might do it by hand, but these systems understand the scalability and the movement of the data. They understand about submitting jobs remotely. They understand about the queues in clusters and how to trick the PBS queues to give you more priority. They understand all of that. They know which queue has less traffic and so on. And they can record what you actually run. So I think workflows capture a lot of knowledge that is missing in the data-intensive science discussions. All the topics that I showed you before. And I think they also help with this issue of reproducibility. So why do we want semantic workflows then? So having workflows, a workflow system in mind, we went off to develop a whole library of workflows for, in particular for population genomics. And this was in collaboration with Chris Mason, who's now at Cornell. He was at Yale at the time. And so this is for population genomics. So for example, on the left are some workflows for association tests. So one of them will do population stratification, and then it does the association test, and then it does some annotations. Another one might use a particular kind of association test, which is a structure association. There's some other workflows for CNV detection. So there's some that use different copy number variant algorithms, and others that combine several algorithms into an ensemble method and then use some combination or voting scheme to combine the results. So different workflows really capture different methods or different approaches to do these population genomics. And what's interesting is that if you're confronted with this library of workflows, what might work better? So for example, there's a lot of knowledge about when these methods are applicable, what data they fit better, and so on. So for example, association tests are better if you have data. That's for people that are not within a family. But if you do have families, you might need to use a TDT test, but only if you have more than a certain threshold, like 100 families. If you have data from the same individual, then you may use the variant discovery from resequencing. So there's different types of data that are more appropriate for certain types of methods. Now, if you hone into particular types of methods, for example, the association test, well, there's one that uses a particular CMH statistic, and you might be interested in only that one. There's also maybe some workflow that will deal with outliers and remove outliers, and others workflows, other methods don't have that. Other times you might, as it says here, another method that incorporates parental phenotype information, and that might be some data that you have. So there's actually a lot of knowledge that we can expose about when these different methods are useful. Here's another workflow that was not working for us until we discovered that actually if there are duplicate individuals, some of the software gets messed up. And so why couldn't we just capture the knowledge and associate that and check for that from the beginning before we run it and fought with it? So what we're doing is taking all of this knowledge and all of these constraints and augmenting workflow systems with that knowledge. So we've created an approach to represent semantic workflows, and we have a system that we've implemented that does this. So every constituent of a workflow, whether it's a data set or a computational step, has a variable associated with it, and we're able to then express constraints that restrict those variables. We also can represent classes of workflow steps. So for example, you might have a step that does classification, and you may have many different algorithms to do that. We have developed algorithms that actually propagate these constraints throughout the structure of the workflow. So the constraint might apply to a particular step, but within the workflow that might have implications for other steps. And the system actually works with web standards or semantic web standards. So we are compatible with a lot of ways that exist today to export and import ontologies and other kinds of knowledge on the web. So let me show you a little bit, in a little bit more detail, what this might look like. And I don't mean to dwell too much into the details of it, but just to give you an idea. So we have ontologies that help us organize data, but also the software that is used by the workflow. So these are examples from text analytics. You might have documents. One of the things that they may have is markup. So you may have markup in them. You may have HTML documents, LaTeX documents. So those create different subcategories of types of documents. But also every document may have a property, which is the language that they're written in. So English or French and so on. So the blue arrow indicates that that document has a metadata property, as we call it. So all of that is represented in an ontology. It's not just your input data, but also any data that's created by the workflow steps. So for example, in doing feature selection, you create a feature vector, and so that has a certain size. So that's also a type of data that's manipulated by the workflow. On the software side, so you may have different modelers, you may have linear regression or a decision tree modeler. These may correspond to different algorithms, so we can start to organize the software components in this way. And then at the bottom, I'm showing different code implementations that may be alternative ways to implement linear regression in MATLAB or R. But we can actually express in the workflows that this step is a linear regression, and then the system will figure out what implementation you have available and map it to that. So we use this knowledge to organize the different workflows. So here's an example where the workflow has these two steps that I magnified. One is doing term weighing on the words in the document, and the other one's doing a correlation scoring. There's actually many ways to do term weighing, and here we've selected one of the ways, which is TF-IDF, and for the correlation scoring, chi-squared. And then on the very right, the actual codes that were run. So we can actually specialize the workflows to be more and more specific until they are executable, but we can actually express the workflows on the left, which really capture a higher level method. So you see the difference between the method that I followed, the variant of the method with those particular algorithms that work with my data, and then what I actually run and executed in case you want to see the details. So this is way too much detail, but just to tell you that we have a language to express what kind of data different components require, how the metadata changes when you execute a component, constraints over the workflow. So we have a language to express the kinds of knowledge that I gave you examples of before for genomics. And what happens is that those constraints apply to particular data sets or particular components. When you link them together in a workflow, we actually propagate those constraints, and that gives us a lot of useful information. So let me walk you through this with a very, very simple example. This is a workflow that normalizes data, takes a random sample, and then it's an ensemble method that applies 3 classifiers and then does a voting scheme to combine the results. Very simple workflow. And there's constraints to the individual algorithms that I'll show you in a minute, but we have algorithms that propagate these constraints, basically doing goal regression, propagating from the results at the bottom back to the input data and then forward. So let me show you really simple. So here's a constraint that says that ID3 modelers, these algorithms, need discrete data. So that means that this input data set will be marked as, with a requirement, a constraint that it needs to be discrete. As the algorithm navigates, it realizes that when you take a random sample of a data set, you're not changing whether it's discrete or not, so the input to that needs to be discrete as well. And when you're normalizing same story, the input to that needs to be discrete as well. So now you know that the input to your workflow needs to be discrete data, because otherwise it's going to break when it comes to the ID3 step. And now we're going to propagate this constraint forward, and going down each steps, which I'm not showing here, it's going to show that the final models will be also applicable to discrete data. And so through the vote classify step, which is not going to change that property, we know that the test data also has to be discrete. So from knowing locally that the ID3 method has that requirement, we've been able to propagate it to now show what kinds of inputs, what kind of constraints this entire method or workflow places on the inputs. So it's a very simple example just to give you a flavor of how the system is reasoning about things for you. Here's another example where I showed you before the earthquake simulation workflow. So the original workflow template just describes what methods you're going to apply, what algorithms, what steps, but of course here comes 74,000 possible fault ruptures. The system's going to reason through them and create individual jobs for each of those fault ruptures. So we have a language to express collections. It's kind of like a very simple version of MapReduce, except you don't have to re-implement your algorithms to run the workflow. You do have to do that for MapReduce. So at the bottom, I show a very, very simple example where the grayed out items, it means that you take a data set, you split it, and then the grayed out area is supposed to repeat it for all the data, and on the right is what the workflow system will generate automatically for you. So, so here's how we use this, for example, to analyze biomedical image data, and this is in collaboration with people at Emory University. So the workflow is shown on the top left, and the system is going to look at some of the parameters of the workflow that will tell us. We have 3D images of a brain, for example, so we have a lot of Z layers, but also for each XY layer, we break it into tiles because it makes the system more efficient. If you break it into tiles that are too small, then the quality of the result will not be as good. So the curve that we show on the bottom is after running the workflow with many different parameter settings, they get a better sense for the quality performance trade-offs, and so they can actually, sorry, the title has a typo. So it's performance quality trade-offs where they can see, if we want an answer within the next day, then we have to live with this kind of quality, while if we have 2 weeks, then we can get this better quality. So that's what I can show you about semantic workflows. I want to show you a bit more about what other things we can get from semantic workflows. So far, I just told you what algorithms we use and how they work, but I want to show you what are the benefits to a scientist. So one is that we can automatically elaborate workflows, and we can generate metadata for the results. Another one is that the methods are more reusable, more readily reusable, and also reproducibility. So let me tell you about these a little bit. So I'll show you this automated elaboration and metadata generation with an example from a workflow for mRNA-seq. So I can't say that I understand a lot of what this workflow is about, but I know that it uses Baguette and BWA. It does alignment with the human genome, a certain build of it, actually. And then it starts with some QC and then runs poly-Baguette, all kinds of different off-the-shelf software. But what's interesting here is that there's a bigger method to the workflow than just the individual software tools that it's using. So this is the very high-level sketch that describes what the workflow, what the method is doing. I don't have the technical skills to walk you through this, but I show it to you so that you get an idea of all the different steps that it's going through. But what's interesting is that then we turn this into a workflow. We know that there's a step, the second one shown there, that is actually, let me see if I can point, splitting the initial samples into many smaller files, trying to process them in parallel. They all have to go through all these steps, and then we merge the outputs and run the rest of the tools. So We have constraints that help us understand how the different steps relate. We start with this workflow template. The system's actually annotating things throughout. It knows how many jobs it needs to split out. It knows the properties of each of the intermediate data sets and so on. And so it's going to generate a workflow that might look more like this, with many individual jobs, how they're related, and then run it, generating the metadata in the meantime. So there's a lot of benefits to being able to track and run workflows of this size. And remember that we started with something much simpler. So that was about the automatic generation and metadata propagation. A second aspect is reuse. So these are some quotes from various documents. I've been working lately on geosciences. So these are, the first one, for example, is a quote from NASA saying that a lot of scientists spend, in their case, 60% of their time just preparing data to fit a model. So this is actually something that they resonate a lot with, that just to set up the baseline for a study for a new project, that takes out most of the time. Also, they talk about quality control. So they say that some groups do invest a lot on quality control, but others do not. And so they would really like to be able to share quality control workflows that are done in the best way possible. They're really chiseled to do things properly. And why can't one reuse that across? Also, people say, you know, I often repeat the work that I know there's 100 other people doing the same thing. So for example, taking data from NASA repositories, the DAC repositories, their satellite data or LiDAR data, and I know that I'm processing in certain ways, and there's 100 people out there doing exactly the same thing. And that's not even my science. That's just getting the data into the format that I can use it. So I think being able to reuse a lot of the, it's not just individual piece of software, but just the methods or a whole entire quality control routine composed of many steps would be very helpful. So we've been looking at this for text analytics. We've been developing a lot of workflows that just combine different ways to analyze text. And what's interesting is that we're finding that, of course, not surprisingly, that once you get features from a document, which are essentially words in the document, the rest of the workflow is really pretty general machine learning stuff. So we've been actually using these portions of these workflows for image and video analysis. But I'll show you in terms of reuse, we've actually found several situations now where researchers that are, you know, very skilled and are trying to do text analytics on their data, they just have text analytics data. In this case, it's a site that has a lot of, collected a lot of question and answers from a scientist network. So they started to download text analytics software, and when they saw these methods that we had, they kind of had this aha moment. Oh, I need to not use all the features. Okay, I get it. So how do I set the threshold? How do I go about this? So it's kind of a way to help them, you know, explore and reuse state-of-the-art methods more efficiently. The workflows often use visualization tools. So on the lower right, there's a way to visualize some of the results. So they also find that useful, that whoever designed the workflow has some particular visualization steps included that help you understand the data. We also had high school kids, so we asked them to, you know, analyze data from the web, you know, whatever they like to do. So they spend just a day trying to learn about workflows. They started trying to classify data from Wikipedia. It sounds very serious, but they really wanted to see if these document classification algorithms could distinguish vegetables from politicians. You know, these kids can be very creative. So they started running the different workflows. They understood what they were doing. They drew that curve on the right-hand side on Excel, and they said, you know, the performance does not go up the more data that we give the system. That's interesting. It also seems to go down. So they kind of started to ask us about that, and they basically discovered overfitting, right? And then in their fourth day, they created the world. In their fourth day, they actually said, we're tired of this Wikipedia data. We want to look at Twitter data. It's like, okay. So you have these topic detection. Oh, I see. They look for words. Let's look at that. So they looked at what topics were trending when the Haiti earthquake occurred. There was a data set for that, and so they could actually see, there's actually lots of different problems with the tweets, and they started pre-processing the tweets because there's a lot of URLs. There's also tweets in different languages, but then the basic method to do the topic detection was there for them, and they could actually use it and get some interesting results. And this is what I was talking about before, that we actually find workflow fragments that apply, and we can create other workflows from them. So this is very interesting that a lot of workflow fragments repeat in various applications. And then the final thing I'll tell you about in terms of benefits, not just reuse of methods, but actually the reproducibility. So here's an example of a paper we've been working with the authors for the last year or so. This is it's called the tuberculosis dragome. It's a paper from Phil Bourne and collaborators, where they actually take all the proteins in the tuberculosis and then look at FDA-approved drugs and try to figure out various ways in which the drugs and the proteins interact. And so they suggest drugs that might be used to treat tuberculosis. So even the abstract says that this method can be applied to other pathogens of interest. It's not uncommon for papers to be really hard to reproduce. So it actually took us 2 months of effort just to reproduce what this method was doing. So the paper was really well written, and we actually were working with the authors, but we realized that it's not just that it's important to have the data sets available, which they did. They actually publish on the website the data sets. It's not important to just have used open source software, which is not all papers use, but they used open source software. It turns out that some of the versions, of course, get improved and get changed and so on. So what really we realized is that because they had kept the intermediate data, because they had kept all the scripts that they used to set up the software, that helped enormously to reproduce the final results. So to us, this is a lesson that the more we expose through capturing all those pieces through a workflow system, not to mention that there were 2 students involved that left the group since they did the work a year ago. So, that involved a lot of phone calls overseas to figure out what they had actually done. So on the right-hand side, we actually created what we call reproducibility maps, which is how hard would it be to reproduce a particular step in the workflow for someone with minimal knowledge about biology, but actually some knowledge about how to set up software, download these, SMAP was one of the tools they used off the web, follow what they're saying. I took these proteins, I ranked them through this, I did that. So someone with minimal knowledge, someone who's a novice, maybe a postdoc that was new to their lab, and then the authors. And so what we found is that you really have to be the author or, equivalent peer, in very tight knowledge of that particular field to really be able to reproduce the results. And in this paper, we actually quantified how much effort that took and why. So here's a grand challenge that would be interesting to me, which is to take these batteries of workflows that are common methods and try to apply them to all the data that exists in the world. Why can't we do that, right? So I'll show you some. You may think this is really hard because every association test study is different and people have to analyze data differently. But here's something very curious. We took a paper that has published an association study, and we took one of our workflows off the shelf and we got the data from the authors. And it took us 5 minutes to do this, but we actually got the same results that the authors got. And the paper that was published actually used proprietary software. They had a very completely different implementation. It's actually good news that we get the same results, right? So my question is, why spend effort and time replicating all the software across all these labs or across the desktop of every researcher within a lab, when you could actually try to reuse these methods. And actually, if we made them more uniform, or if we run these common or standard methods across many data sets, we might get a better understanding of where they break and how they work. And we did this for this other study, which was a copy number analysis. The study, the original study, actually used a very old CNV method. We used a more modern method that was a combination of algorithms, and we got significance in the same gene, the PARC2 gene. And so what's interesting is that, that's good news, right? A more modern method would give you a consistent result, the same result, but it just means that I have some hope that we may be able to apply common methods across batteries of data. And I think, there's going to be an 80-20 rule where 80% of the time we're able to find something pretty quickly. And 20% of the time the data is going to be peculiar or different or really challenge, researchers' intellect in trying to change the method, do something different, see what's wrong with the data. But I think that, maybe it's not 80-20, maybe it's 60-40, maybe it's something else. But I think that it's really worth considering, saving researcher time and trying to reuse these methods and bringing and capturing more knowledge about this data-intensive science. I'm going to skip this and go forward. And just give you this summary. I started out telling you about data-intensive computing, all the things that it focuses on, the scalability, the high-end computing, remote job submission, clouds, what does it mean, all this virtualization. And to me, that doesn't focus on really the human bottlenecks. So people that collect the data and have the data have a really hard time accessing the expertise to data analysis. I think also a lot of the infrastructure that we have in science doesn't allow for reproducibility. I don't know in biomedicine, but at least in geosciences, the use of Excel spreadsheets and errors and all kinds of things is very widespread. I think that being able to capture the knowledge that is involved in analyzing data in as much as possible, the entire end-to-end process as workflows is very valuable. I think that workflow systems can take care of a lot of the mechanics of running these methods, capturing the provenance of the results, annotating metadata. And I think I've also described to you why capturing additional knowledge as constraints in those methods and how the different steps need to be used really help us elaborate higher level workflows, generate metadata for results, reuse methods across domains through defining workflow fragments, and also with reproducible results. You can actually, if you're curious about semantic workflows, you can go to this site, wingsworkflows.org at the bottom, and there's pointers to tutorials, papers, all kinds of things. You can actually go to our workflow portal and run workflows and try them out and have fun with them. And if you want to do more complex stuff, then we can guide you through it. So I'm going to close making a comment more broadly about knowledge infrastructure for science, so an emerging field of discovery informatics. If you think that I talk about semantics and workflows because that's the research that I do and I think they're very important, this is from the EarthCube initiative at the NSF. So it's a 10+ year visionary effort from the community to design cyber infrastructure for the future of geosciences. So that's ocean, atmospheric, and earth sciences. And they talk about, if you look on the left-hand side, they talk about accelerating research. They talk about improving productivity. They talk about knowledge management for science. So I think that these are very much present in the minds of people that are envisioning next generation research. And actually from the EarthCube project, They did a lot of community questionnaires and discussions and workshops. It's a very grassroots effort coordinated by NSF, but their big threads were data, which is not surprising, workflows, semantics and ontologies, and also governance. How do you coordinate and do stewardship of all these different communities and coordination and so on. So, but semantics and workflows as very important topics, and these goals here are very consistent with this message of a more knowledge-driven infrastructure. And if you're interested in the topics of this talk, there's an upcoming symposium in November in Washington, D.C. Just search for Discovery Informatics in the web. There was a Microsoft eScience Summit. There's a meeting at the Pacific Symposium on Biocomputing in January on mass phenotyping around these topics of Discovery Informatics. It's not just about supporting better data analytics, but also how to connect data with models better, and also how to enable more interactions within science, more open science. There was also an NSF workshop, and you can read through the final report. That's very interesting around these themes. This is my last slide. There's a lot of future directions, lots of different areas in which intelligence and knowledge and reasoning can be built into science infrastructure to really bring knowledge more to the forefront. So being able to reuse workflows, assisting scientists with the creation of workflows that are validated in some way by all these constraints that are known to people that use the algorithms every day. Being able to help scientists share workflows, copy and edit them, and so on. And also what I mentioned in one of the last slides, creating batteries of workflows and applying them to all the data catalogs available might be a good grand challenge. So that's the end of my talk, and I'll take questions. Thank you.
How do you view the workflow as being different or similar to coming up with a good API for whatever problem domain you're working on? So let's say an API for machine learning and text analysis, such that ideally you have an open source system that everyone can use and everyone can then distribute simply their code. And that code itself is the mechanism for producing the results. So could you elaborate a little bit on the similarities and differences of those two lines of thought?
Right. So I think they kind of go hand in hand. So when we design the workflows, we try to make the components as reusable as we can, but they need to be specific to the method. So they need to be invoked and be able to treat the data that you need. There's a lot of the steps that use algorithms that have maybe 70 parameters, but only 3 of them are really interesting for this particular workflow or context. So I think that that's part of the design of the workflow. I'm sorry that people have to go, but Alex told me that we had till 4:15, so I was taking a bit more time. Any other questions? Curiosities, comments, thoughts? Not you. Go ahead.
So,
I'll come back to you, sorry.
how much of this can be done with makefiles, careful application makefiles?
All of it. Yeah, if you have the patience.
Yeah.
So the problem is that, so you start writing the make file and you start noticing that you start using bigger data sets, so now things start to fail, so now you need to write more make files or more scripts so that you recover this. Now you want to track how many times you've set this parameter to something, so you start writing this. Now you want to propagate metadata, so if I use data from the Wall Street Journal documents, then the result corresponds to the Wall Street Journal in these dates. So you start basically writing a workflow system. Same thing when I see people using clusters and they start saying, you know, well, you just use these make files, but I'm going to be clever and I'm going to say if these jobs take less than this much time, then I'll group them together and submit them as a mega job to the cluster. That's something that the workflow system would do for you. So there's, you know, a workflow system is a program, so you could certainly replicate it in your lab every time, but it just seems not a very productive way to go about it. Go
So I think this is a fantastic idea, everything you've been talking about today. And I'm wondering, can you talk a little bit about how to make it a reality? What are the feasibility questions that we must consider in trying to see how to deploy this and actually have it?
Mm-hmm. So there's a lot of workflow systems that are deployed and used, right? So for example, we run on top of a system called Pegasus that is used in the back end to a lot of astronomy projects and so on, that does very large-scale data processing. So that's really operational and used. And I mentioned other workflow systems like Galaxy and Gene Pattern, right? So they actually, it's interesting. So the workflow systems that are deployed have very simple capabilities. They don't have these capabilities of being able to reason about the applications or assist a user to create a consistent workflow or anything like that. But they are deployed and used. And the particular plus that they have for scientists is that they do have a battery of algorithms that scientists use every day. And so the workflow system gives you some facilities. So for example, Galaxy gives you a way to connect the data from any step that you execute in Galaxy with the UC genome browser. So that's a nice plus, so I'd rather do that than just download the algorithm and run it locally. So, I'm
really trying to figure out what is that last mile. Do we need to, let's say, for biologists to use this to describe how to reproduce their results in their experiments? Do we need new classes for students? What's the overhead in trying to learn how to use the system? What is that last mile?
So maybe that's a question that I can ask biologists, because I give this talk, people get excited, so why wouldn't you use it? So part of it is maybe a matter of the cost, right? So there has to be a pain point to how much cost it is for you to reproduce workflows in different groups in your same lab. Maybe you want to save in that effort, right? But typically, if I publish a paper and I used a particular method, publishing that and making that available is going to take extra time for me. So how do I get some benefit from doing that? And so I think that there's a lot of discussion about getting credit for people that publish data. There's entire conferences on the topic of data citation. And I think, it's the same issue with these workflows, right? If you talk to people, they don't even acknowledge that these make scripts and Perl scripts that they use really have any value in science. People don't exchange those or publish those, right?
Do you think that
so I think
NIH should require these workflows to be released with each paper? Do you see that as the necessary step for us to go that last mile?
I think that you're going to a very practical recommendation NIH should require. In fact, I don't know if NIH should require. There's many different mechanisms to do this, right? Maybe universities should require that. Maybe publishers should require that. Maybe lab directors should require that. I don't know who could be responsible for enforcing these things, but
what's your guess? What should we do?
So I think this is part of the sociology of science. So I'm not a sociologist of science. I don't know what incentives people in biomedical research have to adopt new technology and to use tools like this. My job is to develop them and to come all the way here to sell them, to tell you that they exist. And so I don't know, what's your impediment not to use this in your work? You might be more qualified to answer that. Maybe there's other people that are thinking about this right now.
So at some point you mentioned some notations for expressing these workflows. I was trying to look through your papers and it wasn't obvious to me if any of them define a language formally and give syntax and semantics. Is there one particular reference of it?
Okay. So there's a paper that we call the Bible, and it's a paper at the Journal of Theoretical and Experimental AI. And so it really goes through a formalism of the workflow representation language, the semantics, and how the algorithms use the constraints. But actually for a user that is not relevant, because we go through a lot of pain to make sure that the structure of the workflow is a very simple data flow language. There's no control constructs. You can never do any kind of programming expressiveness. You can't define variables. You can't define loops. It's very simple data flow language. And so that's why we're able to expose it through this user interface. So you're basically composing a data flow, which is pretty simple. But if, sorry?
It's called the Bible. That's the name of the paper.
No, that's how we call it, sorry.
What's the title?
It's in the Journal of Experimental and Theoretical AI. If you go to my website, or send me email and I'll be happy to send it.
Okay.
And someone else had a question, I think. No? Okay. Oh, it's the guy in the back moving the camera. Okay, thank you.